right, our next and penultimate presenter is Chathura Perera from the Department of Biological Sciences. The presentation title is Pausing by Tangling, How RNA Knots Tell Genes to Stop and Go. Chathura has a very fun fact. My friends and I once got lost in the mountains of Sri Lanka until villagers found us. They were calm, we were not. Guess who led the team? Also, another good one, I'm sorry. I once voiced an international noodle commercial. I gotta find that.
And nobody believed it was me.
Yes, that's...
And nobody did.
Yeah, I kind of, having a hard time believing that. Okay, all right, I believe you. All right, Chathura. Ready, set, pitch.
How many of you have ever taken antibiotics? Almost all of us. And how many of you expect them to work? But for some people, they just don't. They lose even life, not to injury, but to an infection untreatable. This is antibiotic resistance, a crisis affecting millions every year. And we must fight this. For that, we need to understand bacterial control systems down to the smallest detail. And that is exactly what I do. Now imagine this room is a cell and you're inside. Down long roads, tiny vehicles are moving. And as they move, they spin out a long thread called RNA. Like any other vehicle, they pause at molecular red lights. Sometimes they pause even longer. Why? That RNA gets stuck inside as a knot. And this knot is not a mistake. That is how they adjust timing to make better decisions. Every living cell depends on these timed pauses, even dangerous bacteria. The better we understand, the better we change them, right? And here's the puzzle. We know pausing happens, but we don't fully understand why some RNA knots hold the pause longer than others. Right now, scientists study these knots, but one at a time. It is like judging buffalo's weather from one summer photo. And that limits what we understand. And I built a smarter way. Instead of one, my tool studies thousands of knots. Last time, I tested over 65,000 different versions of the same knot in one time. Somebody else would have repeated the test 65,000 times. And that way, we can see how the knot forms and how it slows down the vehicle. And my tool allows scientists to study urgent biological problems, including antibiotic resistance, but not just one case at a time. Of course, I don't make antibiotics. I made a powerful tool to understand principles, so no more guessing. And also, you've seen how apps predict traffic with lots of prior rides. Like that, as I collect more and more data, I can even predict pausing with computers, even without lab experiments. So next time you're waiting at a traffic light, remember, pausing is built-in control. For the first time, we can explain it and predict it, and with that, we are going to change the way how we fight deadly infections. Thank you.